we're going to start. Welcome to our seminar series, Forskning i framkant. And I'm very glad that so many people are here to listen to a very distinguished guest lecturer that we have here. His name is Bud Craig, and he's from University of Arizona, Phoenix, at the Neurological Institute, Barrow Neurological Institute. And he's going to give a lecture on how do you feel, and you have to explain the rest afterwards. But first, Anne-Marie Landblom from the Neurological Department are going to give a more thorough introduction.
And actually I just wanted to wish you welcome on the behalf of the Division of Neurology on the University and also the University Hospital Neurology Clinic, because this is actually the start of your visit here as a guest researcher, very high profile, for 1 year. So we will see you again another 2 or 3 times this year, and we also have some hope for the future that the research that now is started will continue even longer than this first year. So we're really very happy, and these 2 days that you have been here, we already really started up some projects in neurology and neuroimaging together with CMIV. And now Anders Blomqvist, which is your old companion and research fellow, will give some short introduction of the previous science that you've been performed.
Thank you, Anne-Marie. Well, I'm also very glad and very privileged to introduce you, Dr. Bud Craig. And I have known Bud for, I think, 30 years, and this is, I think, one of the great things with science. You get to meet people you would otherwise not have met, and people that become your very deep friends and that it will influence your life. So science has many advantages. Dr. Craig got his Ph.D. degree at Cornell University, working with Daniel Tapper on electrophysiology of somatosensory processing in the spinal cord. And then he moved to a postdoctoral position at Washington University in St. Louis, working with Harold Burton on pain pathways, nociceptive pathways. And from there, he spent another 5 years in Germany with Robert Schmidt's laboratory, first in Kiel and then in Würzburg. And then he was recruited back to the United States to the Barrow Neurological Institute, where he has remained since then. And already during his early postdoctoral work, Dr. Craig described or discovered that pain sensations were processed by separate discrete neurons in the spinal cord and in the brainstem. He showed this in several species, including subhuman primates and humans. And his work on the nociceptive system, I would say, was a paradigm shift. It shifted our view on how the nociceptive system is organized. And from there, Dr. Craig revealed that the nociceptive system was intimately connected, both functionally and structurally, to systems that tells us something about our body, the so-called interoceptive systems, like systems for taste or hunger or thirst, etc. And that the pain system actually was part of an interoceptive sensation or a homeostatic sensation, which of course is very important because homeostatic sensations are influenced by other homeostatic mechanisms, and pain and pain sensation will vary according to homeostasis. And his work on interoception then has led him to formulate a hypothesis also on how interoception actually is a basis for our awareness and our consciousness. And some of these ideas were summarized by him a few years ago in a paper in Nature Reviews Neuroscience. It's called How Do You Feel? And I looked it just up. It has been cited an amazing 108 times this year alone. So it's probably one of the most influential papers published in Nature Reviews Neuroscience. And earlier this year there was, I would say, a follow-up. How do you feel now, in the same journal? And I guess that is what we are going to be told about. And as an answer to this question, how do we feel? Yes, we feel very eager to learn what you will tell us. Please, Bud.
Thank you, Anders. It's a pleasure to be here. Anders has given you an outline of my talk, so now you know what I'm going to say. I hope you are all feeling well. I'm feeling a little chilly. That's why I've got my knees covered with my coat, because they feel cold, and that's affecting me, I'm afraid, because my hands are very cold as well. I live in the desert, as you can see, where it's a little bit warmer. Right now it's probably about 25 degrees Celsius, a little different from here. And as I will tell you, how you are feeling right now will affect everything about you. How you think, what emotions you're having, and how your body behaves. And that's the subject of this talk, because I'm afraid when René Descartes came to Sweden 300 years ago and taught that humans know that they exist because they think, he made a grand step away from what the church had been teaching for thousands of years before that, but he left our brains somewhere off in this metaphysical space, and they really belong in our bodies, because that's what we are. We are biological organisms, and our bodies are what our brains are designed to take care of. So this is how you feel. And this is what textbooks will tell you. Your brain is organized into places that are associated with movement and with touch from the body and with vision. And these are things we know based on wounds to people's brains and upon electrical stimulation to people's brains. And one of the classic studies using electrical stimulation was performed by Wilder Penfield and his colleagues at the Montreal Neurological Institute in the 1950s. And they stimulated in epileptic patients, over 1,000 patients, and when they stimulated here, the patients said they could feel a buzzing in their leg. When they stimulated here, the buzzing was in their fingers, or here in their mouth. And this homunculus, or map of the body, is published in all textbooks. And this is where people believe that we have all of our feelings from the body. And if you note, you'll see that Penfield was able to evoke some visceral sensations over here on the midline, and over here, deep within the lateral sulcus, but he didn't know exactly how they were organized. Now this idea that this somatosensory cortex is the part of your brain that is important for feelings from your body is embedded also in the pain literature. And all textbooks contain a map like this one, which shows that pain and temperature arises in the spinal cord in a contralateral spinothalamic tract that is distinguished immediately from touch because it projects up the other side of the spinal cord. That's the basis for the classical Brown-Séquard syndrome. But then, up here in the brain, it somehow distributes itself back to somatosensory cortex. And all textbooks teach that that's important for pain and temperature, even though it's well known that stimulation of somatosensory cortex almost never causes pain or a feeling of temperature, and a lesion of somatosensory cortex almost never affects pain or temperature. Now this paradox is something that bothered me when I was a student, a graduate student in the 1970s. And I didn't understand why the textbooks would contain a contradiction like this. Of course, I passed all my tests by saying this is the way it is. But that's not the way it is. And this is an experiment I will return to several times. This is an experiment using PET imaging, the positron emission tomography, which is the imaging of radioactive water injected into the body that gets soaked into regions of the brain that are highly active during the 30 seconds that we used to stimulate the hand with cooling stimuli in this case. And this was a technique that was pioneered at the Karolinska Institute here in Sweden. Now, when we imposed cooling stimuli on people's hands, and these are not painful stimuli, but innocuous cooling stimuli that were graded along 6 different temperatures like this, and then performed a regression analysis against temperature across all of the PET scans in these patients, we found that the region of the brain where there was a linear related activation according to temperature was in this position. As you can see, this is not somatosensory cortex. It's not somatosensory 1 or somatosensory 2, which lies right here. It's a region of the brain here that's inside the brain, and it's called the insula because there's an island of tissue that hides deep inside the brain that is normally associated with visceral function. Now we thought, well, that could make sense if temperature sensation was really important for the way the body organizes blood flow. And as a matter of fact, it is, because thermoregulation is controlled by the flow of blood to our skin and to our muscles and our viscera. And thermoregulation is primal for mammals, and in fact, all animals thermoregulate. Even amoeba will go to the right temperature of water that's appropriate for efficient biochemical usage of energy, which is what homeostasis is all about, according to Walter Cannon's teachings of homeostasis in the 1920s. So only from that point of view could we make sense of the fact that innocuous thermosensory discriminative cortex was actually sitting here in a region of the brain associated with the autonomic nervous system and not in the somatosensory cortex. But the question is really, how does it get there? This is the insular cortex in humans. It hides deep inside the brain. It's the hidden lobe of the brain, and as I will show you, it does a lot more than represent temperature. As a matter of fact, from the imaging studies that are available, we know that it does represent visceral sensation. Distension of the rectum or the stomach will cause activation of the insula, as well as this medial region here in the anterior cingulate. But amazingly enough, activation during anger and sadness and happiness and every human emotion that has been imaged in the scanner will cause activation in the same 2 places, in the insula and the anterior cingulate, that are associated with autonomic function. So when Ray Dolan's group here, Winston et al., published an imaging study using functional MRI and asked, do you trust this face? Ralph Adolphs wrote a commentary article, and he used this face rather than the faces that they used in their study, of course. But what was amazing was that they saw activation in the insula again when people saw a face that they didn't trust. And what was amazing was that that activation was lateralized. It was only in the right insula when it was a face they didn't trust, and when it was a face they did trust, it was in the left insula. Thank you very much. I'm glad you like this man. I feel much better. My left insula is very happy. But what the insula does is what we need to know. Now, I am a functional neuroanatomist. I don't believe that the brain is a mystical place. I believe that the brain is evolutionarily and reproducibly well organized, just like human faces. Every one of our brains are different, but there are a lot of common features that we can identify across subjects and use to dissect its function. I use tools like these, microelectrodes used to record from clusters of neurons or single neurons. Here we're recording from neurons in the monkey's trigeminal dorsal horn that were selectively responsive to cooling stimuli, just like I showed you from the PET scan. And these neurons are here retrogradely labeled from the thalamus. They are here immunohistochemically stained for a chemical. Here we have intracellularly injected a single one of those neurons after identifying it as a cooling sensitive neuron. And so we know that every one of those neurons is actually pyramidal shaped with 3 major dendrites. We can identify quantitatively their physiological characteristics, and here are the summed characteristics of almost 100 of those neurons that project from the spinal cord up to the brain in the spinothalamic tract. And then we can make injections of anterograde tracers at the locations of those recording sites and identify their projections within the brain. And that's how I made this map over about 25 years of work, actually. This wasn't easy, but it was worth it, because by this map we can identify a pathway that carries specific representations of first pain, sticking pain, of second pain, burning, heating pain, of cool, of warm, of itch, of muscle ache, and even of sensual touch, a C-fiber activated pathway that's associated with safety and with emotional bonding in mammals, that's carried by this pathway in the lateral spinothalamic tract to these areas within the spinal cord and the brainstem of all mammals. I want to emphasize immediately that the first place that this region projects to, with this region of the spinal cord called lamina 1, is the intermediolateral cell column, which is the locus of spinal autonomic preganglionic neurons that project in autonomic nerves through the sympathetic nervous system to all viscera and muscle and organs of the body. This, in fact, is a spinal reflex arc for somatoautonomic reflexes. And this pathway right here, I want to emphasize because it may be critically important for the kind of pain that patients with multiple sclerosis have. And that is a proposal that I'm making now that we can identify using experiments here in Linköping, because this center has a collection of multiple sclerosis patients that are well identified and that we can associate with lesions in particular regions of the spinal cord that I believe will explain the kind of bilateral burning cold pain that these patients have in their feet. And this may be the homeostatic pathway that is underlying that, because what this represents is the first stage of a homeostatic ladder, an organization in the autonomic nervous system and the homeostatic reflex arc that is joined by parasympathetic afferents here in vagus and glossopharyngeal nerves that relay in the nucleus of the solitary tract and project to every one of these same sites as the lamina 1 spinosympathetic afferent pathway does. And in fact, in primates, both of these regions project to these sites in the thalamus that are selective for this particular pathway. But I have to emphasize immediately that these sites in the thalamus are only present in primates. Subprimates do not have this specific representation of homeostatic afferents in their thalamocortical system. What they have is a representation of homeostatic integration in their brainstem. So the brainstem tells the forebrain of a rat what it needs and how to behave, whereas in primates we have a direct sensory representation of how our body feels. And it projects by way of this specific relay nucleus here that contains a representation of both sympathetic and parasympathetic afferents here to the insular cortex. And I will show you that pathway in a little bit more detail now. First, I want to emphasize that here in the spinal cord, these neurons in lamina 1 represent each of these specific feelings from the body, and they are distinct from the neurons in the deep dorsal horn that actually present some kind of a speedometer, an accurate cumulative input of all sensory input that projects back down to motor neurons in the ventral horn and controls striate muscle. Whereas these neurons here, as I've shown you, project to autonomic nuclei in thoracolumbar cord and control smooth muscle. This is a basic distinction in the spinal cord. There is a superficial dorsal horn that receives small diameter afferent input from all tissues of the body and that is homeostatic in nature, controlling autonomic smooth muscle output, whereas the large diameter myelinated fibers here come around, curl around here, and terminate in the deep dorsal horn, which is controlling striate muscle, skeletal muscle. And these two dorsal horns are developmentally differentiated within the spinal cord in a pattern that was laid down 500 million years ago. This same pattern exists in our cortex, in our forebrain, and that's why I want you to see how old this pattern is. Now, this is an injection of an anterograde tracer that we made in lamina 1 at the site of neurons that we had recorded specifically that were responsive to the feelings from the body, like first pain and cooling and itch and so on. And injections like these allow us to describe the anterograde projections to each of these sites, and in particular to this site here, which is the key relay nucleus for the feelings from the body in primates. And that looks like this. Here's a picture of fluorescent labeled lamina 1 spinothalamic terminations in the thalamus of a monkey. And here in transverse sections in a series across the posterolateral thalamus of a monkey, you can see where the terminations occurred was in a distinct region here that can be identified cytoarchitectonically. It was unfortunately previously identified as a sort of ambiguous zone that was hard to read. And here we can clearly see, guided by this labeling, that there is a distinct nucleus there in the monkey. It is small in the monkey and very hard to see. And that is probably why others before us had difficulty finding that, because they did not have the specific anterograde tracer to guide their observations. We also had double labeling with injections at two different levels of lamina 1. Here the trigeminal level and here the cervical level, and we could see the pattern of organization of the somatotopy of the terminations within this region of the thalamus in VMPO, and that organization is shown here, again in a series of sections from posterior to anterior, here in 3 different monkeys. And here in this monkey in particular, 2 very tiny injections were made in the representation of the face at 2 distinct levels, one near the nose and one near the ear. And as you can see, they are very discrete areas that are anteroposteriorly organized, consistent with this coherent anteroposterior organization here. So there's a map of the body then in a direction that goes across the plane of sections of these coronal sections, and that looks like this. So here we've taken the coronal section and turned it sideways. Those were sections across the thalamus like this, and here we're representing how the body is organized in that projection, and you can see the sympathetic afferents from lamina 1 organized with leg and arm and face in this direction, and then the parasympathetic afferents are organized just in front of that as the body is arranged embryologically. And this, at the junction of the mouth, in fact matches up with the representation in the main somatosensory nucleus of mechanical sensation from touch from the skin and proprioception from muscles, which is organized in a different direction. They are orthogonal to each other, which means they grow along 2 different neurotrophic gradients during development. And that says again that this is one afferent system and this is another afferent system. This is the system that projects the somatosensory cortex. That's what guides skeletal muscle. That's what somatosensory cortex really is. It's sensory motor cortex, because it organizes the input to motor cortex as well to guide striate muscle. And this projects to a different region of the brain, as I've already showed you. Now, first I want to take a little sidestep here and tell you that we can identify in the lamina 1 cells a particular chemical that we call calbindin. And we can identify that immunohistochemically by using antibodies that are selective for calbindin. And here we can double label them and see that calbindin is actually localized in lamina 1 spinothalamic neurons that project in this pathway up to the thalamus of the monkey. And here we can see in a anterogradely labeled lamina 1 spinothalamic terminal that calbindin exists within its boutons as well. So we know then that the terminals of lamina 1 axons in the thalamus contain calbindin, and then we can, in this low power image here, just like the plots that I showed you from the thalamus, we can see that calbindin is clearly associated with the lamina 1 spinothalamic terminals. That means that we can take this marker to another animal, another primate, the human brain, and use calbindin as a marker to identify the lamina 1 spinothalamic termination zone. And that's a project that I did with my friend Anders Blomqvist here 10 years ago in a series of studies that are exemplified here by a complete representation of the calbindin in the VMPO nucleus across all primates. And as you can see, it is very tiny here in the macaque monkey brain, and it is in fact huge within the human thalamus. We found that it was cytoarchitectonically distinguishable here in the human thalamus as a region that was 4 by 4 by 2 millimeters in size. It is immense, and it represents in the human brain this calbindin positive region here, which clearly identified it as a lamina 1 spinothalamic relay nucleus in the human. That is exactly in the site where others have gone into the human brain with a microelectrode and produced, by microstimulation with tiny little currents, distinct feelings of cooling or of pain in awake human patients. We know by making injections at that site in the monkey brain, where we can now make recordings as well, that it projects to this region of the cortex, which is cytoarchitectonically distinguishable and which in fact matches perfectly with the site that I've already showed you, where humans have a representation of linear, discriminative, innocuous cooling sensation. And in fact, it turns out that a lesion of this portion of the human brain, which we now know based on MR identification of such lesions, that this can produce, or even this tiny lesion here, can produce a discrete loss of cooling and of pain sensation from a contralateral region of the body. Now, of course, a lesion of this portion of the brain 100 years ago, made by impact or by a blunt trauma, would have been enough to kill a human. And that's why we didn't know about this in the classical studies of human lesions 100 years ago. We have only learned about it with the advent of imaging studies that allow us to see lesions this deep within the brain. What I skipped over here that I'd like to tell you is that this portion of the brain is activated not just by cooling, as I've already said, but by muscle pain and by laser pain to the skin, as well as deep visceral pain, as well as dynamic exercise, riding on a bicycle, which activates homeostatic afferent input from A delta and C fibers from muscle, whose main job it is to guide the homeostatic nervous system, the autonomic nervous system, in its control of blood flow to muscle and skin. and that's what's happening within this part of the brain, but it represents itch as well and sensual touch, because those are other aspects of the physiological condition of the body, which we can call interoception, that are important for cortical control of the physiological condition of the body, which is now available to primates in a very precise fashion. And in fact, this now provides in the human brain a representation of feelings from the body. The primary encoding in the brain for a feeling sits right here in posterior insula. We know now also that this region is organized in an anteroposterior fashion, here using innocuous cooling of the neck and of the hand, and here using intramuscular injections of hypertonic saline in the leg or the arm, or here in a study done in Göteborg using a brilliant software technique for single subject analysis of sensual touch in normal humans of the leg or the arm. We see an anteroposterior organization here in horizontal sections of this region of the insula, again, confirming the anatomy that we've done in the monkey exists in the human as well. This represents also a method that we hope to use here in Linköping to do some more functional imaging studies of things like cooling and warming that we can use to upgrade this work. That's the second potential project that I've described for you here that we hope to do within the next year. Now, I would be remiss if I only told you that a lesion of this part of the cortex can eliminate pain and temperature sensation, because it is now known that a lesion of this part of the cortex can also produce a paradoxical thalamic pain syndrome, in which a patient complains of paradoxical burning pain in a part of the body in which he has lost temperature sensation, and in many subjects, pain sensation as well. And the primary description of central pain patients is in part due to a neurologist here in Linköping, Goran Leon, with whom I hope to work as well on another project. And that will hope to identify the region in the thalamus that is important for the projection to this part of the cortex. Because as you can see, what was defined by Schmaumann and Leifer in their study of 6 patients who had lesions that produced central pain syndrome, here purely at the cortical level, is the identification of this common region that sits exactly where the cortical area that I've defined for you as interoceptive cortex is. That suggests that if we look in patients that have a thalamic lesion that causes central pain, we would find that it involves VMPO. Now, the basis for this really comes from a piece of work that's 100 years old. Head and Holmes studied 23 central pain patients that they thought was due to a lesion of posterolateral thalamus specifically. And they in fact predicted that this lesion caused a loss of a specific nucleus relaying pain and temperature 100 years ago. And I believe they were right, because what they also said was that what this did was somehow release an area in medial thalamus that produced the ongoing pain that was characteristic in these patients. So the loss of a discriminative pain and temperature region here somehow produced a release of a burning pain caused by activity in the medial thalamus. And that in fact fits with this modern theory that is in fact an extension of Head and Holmes' theory, and that can explain as well the production of central pain by a loss of this region, because what it does is project down here to the brainstem and modulate the homeostatic integration within the brainstem, and those areas project to this region of medial thalamus that goes to this area of limbic motor cortex, which in fact communicates with an area here in right anterior insula where humans actually engender their own feelings. And I will tell you about that in just a moment. So this is one of the two studies in the literature who have attempted to identify the location of a thalamic lesion that produced central pain syndrome, and they determined that that lesion did not involve the lesion that the region that Anders Blomqvist and I described as VMPO, but I would maintain that they didn't reconstruct the thalamus very well with their cytoarchitectonic drawings here, because when I use our map and match it up with what they showed with their MRI sections, I would say that VMPO is in fact included in that infarct and was damaged greatly. And what we need is a new series of studies with high-resolution MR structural images that could be obtained here in the imaging center, where a new technique affords an enormous increase in structural resolution. And with that, we hope to be able to obtain the answer to this question definitively, thanks to Anders' person. So this pathway, I would maintain, is important in humans for the generation of those feelings. And in fact, this pathway has a medial branch that generates an input here to what I would call limbic motor cortex in relationship to a region here that I would call limbic sensory cortex, because this area in fact modulates the sensory integration within the brainstem of homeostatic afferent input, and this region of cortex here modulates this region, which is the motor efferent integration of homeostatic activity within the brainstem. And the way that these two areas talk to each other is now what we really need to understand. And that is what we asked in this experiment again. Because we used PET, we had a long stimulus, and we were using awake human subjects who could tell us how these stimuli felt. So we not only could use the temperature of the stimulus as a regressor to study the blood flow within these subjects' brains, but we could use their feelings as a regressor to study blood flow within their brains, to see what part of their brains was active in relationship with their feelings. And as you can see here in these axial slices from superior to inferior, relationship to the temporal, the temperature regression here, you can see that one site that I've shown you already on the contralateral dorsal-posterior insula. But when we look in this row and we look at the regression analysis against the subjective ratings of coolness that these subjects gave, there is obviously a difference. And there's another region over here in the brain on the other side that is very strongly activated, that is very poorly activated here when we do a regression analysis against temperature. And now I'm going to take you down into the data right here, because I'm an anatomist, and I want to look in detail and see if I can identify the pathway. So here what we're looking at are two different Z levels of activation here. And in the temperature regression, you can see here distinctly the area in contralateral dorsal-posterior insula that I've already told you is the representation of objective temperature in interoceptive cortex. But now when we go to the rating regression, you can see that that area diminishes. And an area just in front of it lights up, as well as an area here that's much larger on the right anterior insula, and orbital frontal cortices that it is large enough to contain several re-representations. And this immediate re-representation here in the middle portion of the insula makes a lot of anatomical sense because it's just like every other sensory pathway, which has a V1 and a V2 for vision, an A1 and an A2 for the auditory pathway, an S1 and an S2 for the somatosensory pathway. So there's an immediate re-representation of interoceptive space in the middle portion of the insula that is somehow already abstracted because it has a closer relationship to subjective ratings than it does to the objective temperature. And that becomes then re-represented and very strongly related to subjective ratings. And then over here is where we want to look for the part of the brain that's important for how you feel. And what's amazing is that that same portion of the brain, I show you here again in a highlighted map, is active in every study of feelings that humans have. Whether it's a feeling from the body, like cooling or pain, or whether it's a feeling like an emotion aroused by watching a sexual movie, or here watching a movie that makes you angry, or here looking at faces that you trust or don't trust, or looking at faces that make you feel disgust, or even faces that make you think the person you're looking at is happy. That's a secondary emotion of empathic happiness, or empathic sadness, or empathic pain, or here even the feeling of being excluded from a game that the computer is playing. And this social exclusion they saw was the same region as this. It's obviously a little more anterior, but there's an area here in the right anterior insula and orbital frontal regions that is commonly activated in all these emotions and feelings from the body, which is exactly what William James predicted 100 years ago in the James Lange theory of emotion, which said that our emotions are based upon the homeostatic representation of our body's feelings and reactions to the stimuli around us. So when the bear is rushing at us, we become afraid because our pupils are dilated and our blood pressure is up and our heart is racing and we feel that, and that's what makes us afraid. Well, this is exactly what's happening. There is a re-representation of your body's condition here that is the same space of the brain that is used for all your feelings. That is the same as Damasio's somatic marker hypothesis in his book, The Feeling of What Happens, and Descartes' Error. This model that I've shown you so far suggests that there is an interoceptive map of the body that is re-represented in the middle of the insula and that is associated with representations of other sensory environments, sensory signals from our environment and from our exteroceptive motor space and with our homeostatic motor system, and that becomes re-represented in the anterior insula, and that is associated with activity in our other portions of our brain that are associated with social conditions and motivations and so on. And this fits with the fact that this region is actually representing a homeostatic integration of the condition of the body and the brain. The brain uses 25% of the energy budget of the entire body, and that means that if this is to be a homeostatically true and faithful map, it needs to represent not only what's happening in the body, but what's happening in the brain as well, in order to represent an energy efficient model of what my body needs. And that's what this model seems to suggest. Now, that was what I wrote about in the first review that Anders mentioned in Nature Reviews in 2002. And the new one incorporated a ton of imaging studies that have appeared in the last few years. And the basis now seems to suggest that this anterior insula and anterior cingulate representation is a representation not only of how we feel, but of how we feel anything. That is to say, our awareness of ourselves and our moment and of others and of our environment, of human consciousness itself. And I can spend minutes talking about each of these studies. I want to emphasize that this is a selective area of the brain that's involved in time perception, selective area of the brain that's involved in feelings of what's going on in our brain while we're trying to remember something, and it is selectively involved in that absolute moment of recognition when something becomes clear to us in our perceptive space. And if I'm going to get to the end in 15 minutes, I need to speed up. So there are a number of human lesion studies that provide strong support for the hypothesis that this area of the brain is involved in awareness. The most important one are studies of frontotemporal dementia, a dementia that's like Alzheimer's, except that the regions of the brain that degenerate are the anterior cingulate and the anterior insular cortex. And in fact, in a landmark study, Bill Seeley and others from University of California, San Francisco reported that the region that contains these von Economo neurons, which are selectively associated with anterior cingulate and anterior insular cortex in hominid primates in a transient phylogenetic progression, which are only present in large numbers in human adults, are exactly the neurons that degenerate in association with the loss of self-awareness and self-conscious behaviors in frontotemporal dementia, suggesting that in fact this is part of the network that underlies human awareness. That is this study here, which they compared and contrasted with degeneration in Alzheimer's disease, because these neurons do not degenerate in Alzheimer's disease, and Alzheimer's patients lose their cognitive function but do not lose their emotional awareness of themselves, and patients with frontotemporal dementia lose the awareness of themselves and of others and of their emotions and of their sense of time. So that suggests a new model, a model in which there is a primary interoceptive template that generates feelings from the body, that integrates with homeostatic motor function in the middle of the insula and proceeds to the anterior insula to a representation of all feelings, including all regions of the brain that are associated with feelings that we can have. And that means this is a representation of me, of the sentient self at this moment in time, one moment in time, and that's all it takes to represent time, is a representation of a number of these across time. Like this. So if you multiply these and reproduce them, and you have a timer that runs across time like this, then this produces a cinemascopic view of the self, that can explain our representation of a sentient moment, a prescient moment in which we exist, which lasts about 40 seconds, and that runs in one direction. And in fact, it runs in an emotional fashion, because if we're falling down the stairs or jumping out of an airplane, that time begins to stand still. It runs very slowly because that time contains an enormous amount of salience, and that can be explained by the fact that these only contain so much information. And if it's a very salient condition you're in, that means you need a greater number of these to represent what's going on right now, and that would mean that time stands still. And that's another experiment that we'd like to perform here. We hope, and if it works, if I can actually make it work. Okay, I think you could all see that the ball that got larger seemed to act like it lasted longer than the other balls. And the sound that was with it seemed to sound like it lasted longer than the other sounds. And it didn't. They were all exactly the same length of time. This is a simple demonstration of the fact that in your brain there is a subjective timer, and time is in fact subjective to you. It's not absolute time that you perceive, and that it can be manipulated in the scanner. And that's another experiment. That's the third experiment I want to tell you about, that I hope to do here with colleagues at the University of Linköping in the next year. If we can perform an imaging study of that illusion here and identify, in fact, which portion of the brain is important for that, I'm sure it's the anterior insula. I hope to be able to show a dichotomy between the two sides. I'm going to skip over this one because it's the dichotomy that I want to get to. This is really important because it is well known from stroke patients that if you have a patient who comes into the clinic who's had a stroke, who is pathologically happy. He's had a stroke and he can't move his right arm and his right leg, but he's really happy about it. He's definitely got a stroke in the right side of his brain. And it's the left side of his brain that's very happy. And if he's pathologically sad and depressed, he's had a stroke in the left side. Is this not true? If you do the WADA test and inject amytal up a carotid artery, the same thing happens. If you anesthetize one side of the brain and leave the other side alive, if it's the right side that's alive, people get depressed. If it's the left side that's still awake, then people get really happy. And this fits with a lot of psychophysiological literature that I'm not going to go through, which suggests that the left side is your happy side and the right side of your brain is the depressed side. Well, that would fit with a homeostatic integration model that suggests that the right side of your brain contains a representation of sympathetic afferent stimuli like temperature or like pain, and that the left side contains a representation of parasympathetic afferent stimuli like food and chocolate and happy music and sex. And it turns out that that's exactly what's in the literature, because the right side of the brain is activated by challenging stimuli, like pain. And here's recognizing oneself in a mirror, if there's not a greater emotional challenge. And here, activating the left side is listening to pleasant music, or pleasant rhythms, or mothers looking at their babies, or listening to happy voices, or making a smile. Activates your left anterior insula. So we really do have this dichotomy in the two sides of the brain. There's a happy side and there's an unhappy side. And that fits with the idea that the two sides actually interact with each other in this classic opponent fashion, which is how muscles work across joints, which is how the autonomic nervous system works in the gut or in the heart. One side speeds it up, the other side slows it down. And as a matter of fact, that's how it works across all vertebrates. Because one side of the brain in frogs is associated with feeding behavior, and the other side is associated with predation. And this was now described very recently in a Scientific American article that summarizes 24 years of work on these very subtle behavioral differences in vertebrates. This is old. This is really old. And what it suggests is that in the human brain, we have an afferent representation that has been guided to the other side to match a pre-existing efferent lateralization, because that is efficient. And that's what homeostasis is all about. And that's what this brain integration is all about, because the efficient use of energy is what biology is organized to perform. Now, one of the things that this predicts is that if you want to get rid of pain, then you want to make people happy. One of the ways of doing that is to stimulate the vagus nerve. People know that stimulation of the left vagus can reduce epileptic seizures, and some people think that it can also reduce treatment-resistant depression. What happens when you stimulate the left vagus nerve is that you activate the left insula. What's amazing is that it causes deactivation of the right insula, exactly as this theory predicts. So what we decided to do was to test this theory very simply, and that was by asking people to breathe slowly. Which activates bronchiopulmonary afferents, which are entirely vagal and parasympathetic. So breathing slowly increases heart rate variability, improves cardiac morbidity, and causes a measurable decrease in pain sensitivity, exactly as we would predict. Well, this can explain why depressed people eat, or why eating chocolate makes you feel good, and a lot of things. In fact, this could be used as a biomarker for emotional dysfunction. And that brings me to another experiment that I hope to perform here in Linköping. This is the last one, and my final slide, because it appears in a paper that is now in press that I wrote with a Swiss neurologist who was fortunate to obtain a SPECT imaging study of an epileptic patient who was experiencing a so-called ecstatic seizure, like Dostoevsky experienced, in which a patient feels unbelievably happy and really well and really aware of oneself, and as if time doesn't exist, and as if the whole world is a symphony of good music. And there was activation of the anterior insula. And in fact, a neurologist here at this institution, Anne-Marie Landblom, reported a similar SPECT imaging study and showed that there was hyperactivation here that she identified in the parietal cortex. What I would show is that there was, in this patient, again, hyperactivation of the insula. And hopefully, her colleague, Brodkorb, a neurologist from Norway, will come and we will obtain some of these ecstatic seizure patients and be fortunate enough to perform a similar imaging study here and really investigate this theory in detail. Thank you very much.
So what happens to people who are, say, sociopaths and don't have anything?
Well, that's a great question. There are, as far as I know, few or none imaging studies of sociopaths in a pathological condition. There is, however, an imaging study of schizophrenics who have auditory hallucinations. They feel like there is a voice in space yelling at them, some disembodied voice that is vulgar and terribly angry. And in fact, those patients have a hyperactive right anterior insula, in stark contrast to the fact that when they're in normal condition and performing a verbal control task in the scanner, it's their left anterior insula that is more active. Okay? Yeah.
So can we compare that most of the psychological problems are due to insula problems?
Well, that we don't know yet. There are several studies that are suggestive. For example, people are interested in relating autism to this region of the brain. There is one good morphological study that suggests that people who have autism and can't relate to other people's feelings have a smaller insula than other folks do. There's another study that suggests that patients with borderline personality disorder, adolescents who have conduct misbehavior, have a smaller portion in the specific region of the insula, in fact, than normal patients, and so on. But this is just beginning.
What kind of transmitter substances do you find in these? Are they mainly GABAergic or what?
Great question. This is one of the areas of enormous density of opiate receptors, for example, in the brain. And this may be one of the regions in which it has its analgesic effects as well as its psychic effects, because there is a study of a South American psychic agent, a hallucinogenic agent like LSD, called ayahuasca, which generates a hallucinogenic happy state, and that causes, they've done imaging to show that that causes activation of the insular cortex. But there's strong serotonergic and dopaminergic input to this region as well. And so there's a lot that needs to be done.
I have an associated question to that. In this country, we are very interested in the intoxicating effects of alcohol. And as you all know, under alcohol intoxication, time perception changes. Could you comment on that?
I wish I could. You know, I'm not aware of anyone who has done an imaging study of drunk people subjected to a timing test, but that has just begun. There are a number of studies of subjective timing in normal people, and as I showed you in one of the slides already, the anterior insula, the dorsal putamen, and the left posterior parietal cortexes are the three areas of the forebrain that are crucial for subjective time perception. So one might guess that there's an effect there. Yes?
So in the imaging pictures, you show that the insula is activated and a part of the cortex, I forget the name of it, but the images indicated that it's not the same exact area that's activated, so there must be a pretty precise map to this. Well, how would you go about sort of deciphering the whole flow of information from the sensory neurons all the way into different parts of the insula, different parts of the cortex?
Well, you know, it's like now we know where the new world is, and now we need to begin mapping it, and that's what's coming next. So there are only a couple studies so far that have used multiple tests to identify the overlap or the distinction between regions in the anterior insula that are activated. Those kinds of studies are crucial now to be able to dissect this region, but that has just begun.
Okay, and then we thank Dr. Craig again for this eminent lecture. Thank you very much. And also as a token of gratitude and optimism for this next year and perhaps more years, Gösta Berlin, who is a representative of the County Council, and me representing the faculty, would like to hand over a book in Swedish, and a tie which is international, which is our faculty tie, and also a T-shirt which might give you a bit more warmth.
Yeah, I hope so. Thank you very much.
Thank you so much.